mitosis, not meiosis, and then later on we'll talk about meiosis. So imagine you have a sperm, there's a chromosome and an X, and these are going to get fertilized and produce an embryo. Now at this point, all of the genes in this embryo are poised to get replicated. There should be two terms of the embryo, obviously. As this embryo divides, there is unequal division, such that some cells will go down a particular developmental path and turn into things like the ectoderm, the skin cells, etc. Others may go down a different pathway and end up turning into your mesoderm or your endoderm, which ultimately give rise to the digestive tract. Now, this stage where a particular cell takes a particular path is known as cell commitment. That cell is committed to that one path. Once it makes that decision, it's going to stay along that path. It doesn't usually go back. Now in the lab, we can manipulate it to go back. But in biology, in nature, this is a one-way path. And so at this point, a certain set of genes get activated in this cell, but not in this cell. And a different set of genes are activated in this cell, but not in this cell. The rest of the genes that are not activated are silenced or inactivated. So there is a commitment stage at the level of the genes that determines the appearance and the trait of the cell. Now once that decision is made, the cell continues dividing, but it inherits its state. It passes it on to its daughter cells. Once a state is determined, it keeps and it inherits that state. And this aspect of inheritance of a particular cell state is known as epigenetics. Each of these cells have all of the same genes. Whether you are an orange cell or a yellow cell, you have the same genes. But the yellow cell only activates a subset of genes. And once it activates that subset, it maintains that subset and not the other. That's known as epigenetics. It's the alteration. So active genes maintain activity, the inactive genes remain silent, very stable across many, many generations. Of these cells. Okay, so how does this happen? Well, you remember DNA is not naked in the cell. It's packaged with histones to form nucleosomes. So here is my cartoon representation of DNA. Here is my cartoon representation of a gene and its promoter. Right? Transcription is going to go from left to right. And it's going to make an RNA across this green box. And that RNA will get made into a protein. That protein will basically determine the fate of that cell. Either it's an exosome or a mesosome or an endosome. So all of the genes are not active in all of the cells. There are certain genes that are developmentally regulated, tissue specific, and that will be activated in particular tissues. And the mechanism by which this happens is by the presence of specific transcription factors or transcription activators. So there are specific transcription activators present in the ectoderm cells that are missing in the endoderm cell, and there are totally different transcription activators present in the endoderm cell that are missing in the ectoderm cell. These transcription activators will bind to the promoters or enhancers of only a set of genes. Those genes become active. The other genes are silent. So, what happens is, to activate a gene, you need a specific transcription activator. And that transcription activator is missing in cells where those genes are not. There's a decision made which transcription activator is made in which cells. Here is a picture of some of the transcription activators that are present that allow a cell to become a specific type of cell. I've taken it from a review 2 years ago. So, when you go to development, fibroblasts get converted to muscle cells. And the key transcription activator that is required to go from fibroblasts to muscle is a transcription activator called MyoE. This activator will bind the enhancers of genes that are active in muscle muscle cells, but not in genes that are active in other cells, such as macrophages or any other. Similarly, there are specific transcription factors that allow a B cell to be converted into a macrophage, and they are called transcription activators CF, alpha and CF, and so on. So each different lineage of cells that you have in your body are determined by specific activators that are made in those cells rather than other cells. And so that's one mechanism by which you go down a particular pathway.
For the fibroblasts going to the pluripotent stem cell, there's multiple ones. Do they all attach to the enhancer, or all of those can attach and produce the pluripotent stem cell?
So to convert a fibroblast to a pluripotent stem cell is only done in the lab. Okay? And to make that conversion from fibroblasts to stem cells, you have to express these 4 transcription factors simultaneously. This one is an artificial system. This allows you to take any cell, or not almost any cells of your body can be taken, and then when you activate these 4 transcription factors in those cells artificially, you can convert any cell back to a stem cell, and then that stem cell can then again go back and develop into other tissues. But this is an artificial system in the lab. It doesn't happen in vivo, or at least not that we've discovered so far. Okay, so here's just a simple pathway of how you get your different red blood cells and white blood cells. You can see how there is an amazing divergence of factors, and there are specific transcription factors that determine at each stage what goes on. This is during the production of blood cells, all the different blood cells, all the way from red blood cells or erythrocytes down to the macrophages that make platelets, mast cells, these are your white blood cells, etc. And actually there is somebody in UCSC who works on this pathway by the name of Camilla Forsberg in the DME department. She focuses on these different pathways and the transcription factors and how they lead one cell type to go down one pathway but not another. So you can see how this might work. So gene activation, you need an enhancer to activate the transcription of a gene. And there are specific proteins, transcription activators, that bind to those specific enhancers and activate the gene. Remember what we talked about in our very first lecture, eons ago, where to activate transcription, you need a protein to bind the promoter or the enhancer, which then leads to the recruitment of RNA polymerase and the general transcription factors, which then leads to transcription of a gene. So these proteins that bind the enhancer determine what the fate of that cell will be. Okay. Now, how do you get cell-specific activation? So this is a hypothetical situation. Let's imagine you have a hepatocyte, a liver cell. This cell has 4 different genes in it. Gene 1, gene 2, gene 3, gene 4. These 2 genes are active in this liver cell. These 2 genes are inactive in this. Remember, the DNA is actually packaged into nucleosomes. The function of the activators is to get rid of the nucleosomes. So it actually removes the nucleosomes from the promoter and enhancer of the gene. And the activator that results in the formation of hepatocytes is a specific protein transcription activator called HLF3. This protein will basically bind a particular DNA sequence. And most importantly, that particular DNA sequence that it binds is like a restriction enzyme. It recognizes a particular DNA sequence and it binds it. But those sequences are only found at the enhancers of genes that are active in liver cells. Those DNA sequences are not found in the enhancers of genes that are active in, say, brain cells. So you have specific DNA sequences upstream of the gene at the enhancer. Those are recognized by HLF. They will bind it, they will remove the nucleosomes, and they will allow the gene to be made into RNA and then into protein. And those proteins will basically indicate to the cell that that's the liver cell. On the other hand, in brain cells or neurons, these 2 genes are active, the brain genes, but these 2 genes are inactive. action activator, NZF2, that will bind to a different DNA sequence. And those sequences are only present upstream of the genes that are active in brain cells. So it's the sequences on the enhancers that will determine which genes become active in which cell type. Because the transcription factors will only bind those genes and not other genes, and turn on those genes. So the activators are really like little switches, and you're turning on or off a switch. That's what these activators are doing. They're basically removing nucleosomes. Once the nucleosomes have been removed, you can make RNA. So that's what the function of this is.
So then what determines what activators are in the cell?
Very big question. So there is what's known as unequal cell division. During these pathways that are taking place, certain factors are unequally divided. There is actually, in yeast, it's been studied in really great detail. You may imagine yeast as being a single cell organism, but there is a mother cell and a daughter cell. And there is a transcription factor that is transported from the mother cell to the daughter cell along little tramlines of actin and myosin, which make the daughter cell have more of this protein and the mother cell have less of this protein. And that then tells the cell, here's a daughter cell, here's a mother cell. It's a phenomenally interesting story to study. We worked on that with UCSF and Europe. And that's how we go down those pathways, basically. So there is unequal division there. Okay, so we talked about specific genes being activated. Certain set of genes are activated here, certain set of genes are activated here. But what about the other genes? The rest of the genes are silent in this cell or this cell. You only activate a small group of genes, the rest of the genes are silent. And how do you maintain this silencing? Well, you maintain this silencing by many different mechanisms. And it's those mechanisms that we're going to be most interested in. One way is, of course, absence of a transcription factor, just as I told you. But there are additional mechanisms that maintain those genes in a silent state. One is changes in the structure of the nucleosomes. And I won't talk about those that it's more complicated. If you take MCD biology, and if you do eukaryotic molecular biology, you will hear a lot of details about these protein changes in the histone molecules. What we will focus on in today's class are changes in the DNA itself. The DNA is not just ACGE, it's modified. Little methyl groups are added at specific sites along the DNA that have consequences for transcription. And we'll talk about that later on in this course. So, gene repression during development is as important as the presence of specific transcription factors in a particular cell type. All genes are not active in all of the cells. And basically, there are also proteins that will actively repress the genes that are not active. Okay? So you need specific activators to activate genes, but you also need specific repressors to maintain the inactive genes in the repressed state. There is a mechanism by which this works, and these repressors are as important as the active genes. So, to have an active gene, you need a specific transcription activator. If you're inactive, you don't have that activator. But you also need repressors. Specific repressors are present that will maintain the inactive gene in the inactive state and will allow the active gene to stay active. They're just as specific as the active genes. And there are specific modifications of the histones that are present in the active genes, but not on the inactive genes. I will talk about them today. There are specific histone modifications that are present on the inactive genes, but not on the active genes. And then the final important one is methylation of DNA. This is DNA itself that is being modified by putting on a methyl group, CH3, right? And that leads to the inactivity of a gene. If this modification is present on a gene, the gene is inactive. If the modification is absent, the gene is usually. So cell-specific repression, let's look at those same cells, our hypothetical liver cell and our hypothetical brain cell. What you find is that the genes that are inactive are coated with specific repressors. Of course, you need the activators as well to activate the liver genes. But the rest of the genome is covered up by other proteins called repressors. In the brain cells, different sets of repressors, different sets of genes are inactivated, basically, and you have specific. So it's a dual mechanism. Repression of genes that are not required, activation of genes that are required. Both mechanisms have to work together to give you proper regulation of genes. If one mechanism is messed up, you lose the fate of a cell. The probability by which a cell divides and gives rise to the same genotype or same phenotype gets messed up when you mutagenize some of these things. So you have defects in the development. Okay. So gene silencing is very important. This repression of genes via these repressors, these little red caps that I've shown over here, is very important as well. Because remember, the vast majority of the genes in your genome are silent in any cell. It's only a small subset of genes that are active in any particular cell. The rest of them are all inactive. Okay. So the vast majority of the genome has to be shut down or be silent. And only a small subset of genes will bind specific transcription activators. Now, DNA methylation is an important mechanism to maintain genes in a silent state. So let's imagine you have these genes. This gene is active, this gene is inactive in this cell. The mechanism by which you inactivate these genes is just as important as the mechanism by which you activate the receptors. Okay, so this inactive genes that you have, the vast majority of the genome, as I said, is inactive. If you look at an electron micrograph of a nucleus, you can visualize it quite clearly. This dark stained region here, along the periphery of the nucleus, and in the center of the nucleus, this darkly stained region, these are all inactive genes. They're called heterochromatin because they're stained dark. And the active genes are this little open region. You can just imagine, to transcribe and make RNA, you have to open the DNA. You have to make it more accessible, more friendly. And so you open it up in the middle of the nucleus. So that's all open and things can gain access to them and transcribe. While things that you want to have inactive, you kind of squish together in a very tightly packaged region, basically condense them. And because they're so tightly packaged together, when you stain them, they look very dark. Now there are two types of inactive chromatin in the cell. Something that's constitutive, that means it's always inactive, never becomes active. And most of this constitutive heterochromatin is repetitive DNA. Remember I told you, repetitive DNA is present throughout the genome. That's what we use to study microsatellites and mini-satellites. But they expand and contract all the time during meiosis. The cell doesn't like that. So it tries to prevent recombination from taking place by packaging the DNA, the repetitive DNA, with these repressor proteins. So the repressor proteins do two functions. They prevent any genes that are present from being made into RNA. Same time, they prevent recombination during meiosis or even mitosis. So they inhibit recombination, they inhibit transcription, they even inhibit DNA repair to some extent. They just basically compact everything. And in the process of compacting everything, everything becomes inaccessible. Recombination, transcription, repair. Any DNA-based process becomes inaccessible. And so this is known as constitutive heterochromatin. It's always inert. It's always repressed. Nothing changes. Nothing is okay. And on the chromosome, most of this constitutive heterochromatin are the telomeres, ends of your chromosomes, and your centromere. Most of the repetitive DNA is present over here and here, and most of that DNA is in this recombinationally inert chromatin state. if you analyze them, you'll find that they are at the periphery of the nucleus. Okay. A second part of euchromatin is the active chromatin. Right? So that's a term we use just to reflect the active chromatin. And then we have what's known as facultative heterochromatin. Facultative heterochromatin is basically inactive in one cell, but active in another cell. So those are the genes that are active in one cell type, but inactive in the other. And the inactive cells, or the genes, are basically referred to as facultative. That's the chromatin that is repressed only in particular cells. So, regions of the genome that are active in one type of cell, inactive in another type of cell. And so we're going to talk about this kind of chromatin today for the next, I guess, half hour or so before we wrap up this class. And this is known as facultative heterochromatin. These are regions of the genome that are rich in genes. There are genes present here, but these are condensed and transcriptionally repressed. And one classic example of this is inactivation of X chromosomes in mammals. It's also referred to as dosage compensation. All right, so let's look at mammals. Okay, we're focusing now on mammals. In the general term, we have two X chromosomes, or an X and a Y, right? So, this is a female, this is a male. Now, if I were to look at the genes that are present on the X chromosome, right? I'm just looking at the genes that are present on the X chromosome. They're going to make RNA. So I'm going to measure the RNA from the X chromosomes in a female and measure the amount of RNA from the X chromosomes in the male. Now, because there are two copies of those genes in females and one copy in males, theoretically, you should expect twice as much RNA for those genes in females as in males, correct? Kind of makes sense? Because you have two copies. Each of those copies gets made into RNA, so you should have twice as much RNA in females for the genes on the X chromosome compared to the genes in the male from that one X chromosome. But when you do that measurement, what you find is that the number of transcripts, the amount of RNA made from those genes from the X chromosome are more or less equal in males and females. And that's a phenomenon that we refer to as dosage compensation. We are somehow compensating for the difference in number. Right? So how can we do this? Think about how you can adjust the levels of the RNA from the 2X and the 1X such that they are equal. How many different ways can you think of? Let's give it a shot. Tell me a little bit about it. What would you do to compensate the levels of RNA? Any ideas?
One of the pair of X chromosomes could be completely silent.
Okay. One of the X chromosome pairs could be completely inactive. That's one mechanism. Any other mechanism anybody can think of? Yes.
You double the transcription of
you
the.
can double the transcript of the RNA from the male. That's another mechanism. Third mechanism is you have kind of approximately uneven transcription. You have less transcription from all of the genes in the female and more transcription from all of the genes in the male. That's another way. So there are three different ways in which you can do this. Let's look at how it works. In flies, the second method is the one that works. In the females, the genes from both X chromosomes are made into RNA. But the X chromosome in males makes many more copies from the same version. So you're making twice as many transcripts from the X chromosome. In flies. In worms, you have a very similar mechanism as well. Worms don't have a Y chromosome, but they have a similar mechanism. Mammals, on the other hand, what happens is one of the X chromosomes is active. All the genes on that one X chromosome will make RNA. The second X chromosome is completely repressed. It's just shut down. almost no transcription from the genes on the second X chromosome. The male's single X chromosome, you get transcription. And so you kind of equalize the amount of RNA that's produced in the males and the females. Okay, so how is this X chromosome inactivated? And what are the consequences of this on specific gene traits or genes that are present on the X chromosome? And you've studied those genes. You've studied color blindness. It's a gene on the X chromosome. You've studied hemophilia. It's a gene on the X chromosome. But what happens if you have this phenomenon taking place on the X chromosome? Okay. Okay, so mammalian males and females have 1 and 2 X chromosomes. And so one would expect that for X-linked genes, you will produce twice as much RNA in females as in males, but that's not the case. And this is dosage compensation. And it's done by inactivating an entire X chromosome in the female. And when you inactivate this X chromosome in the female, you can actually observe it. You've probably seen pictures of this. It's called a Barr body, B-A-R-R. It's what you observe in females, in the cells of a female. One of the X chromosomes is inactive, becomes very highly condensed, and drops together and forms this little structure in the nucleus that you can very easily visualize in the microscope. And the reason for that is it gets packaged with protein, with vessels, to form heterochromatin. And this is facultative heterochromatin. This is heterochromatin that inactivates the entire X chromosome, almost completely. So dosage compensation in mammalian females occurs by shutting off most of the genes on one X chromosome.
Question?
Yes.
Is it the same X chromosome?
Very good question. We'll come to that. Hold on. Right. This inactivation is random. So it happens on either one X chromosome or another X chromosome. It doesn't matter. It's totally random. At a particular stage in development, that does have consequences, as you will see in today's class. Okay. So for a given cell, as the organism is developing, there is a probability that either the maternal or the paternal X chromosome will be inactivated. But once it's inactivated, it stays stable and it is inherited throughout the rest of your life in those cells. And it's because of this stable inheritance in the state, once it's established, you get food. So, here is what happens. Here is a male, X and a Y. Here is a female, XX. This X chromosome is inactive in the female. This X chromosome is active in the female. So I've drawn the inactive X as a kind of little bar body here. As this female is going to make gametes during meiosis, this X chromosome becomes active. Only during meiosis in the ovary of the female, the X chromosome will become active. Okay? So now she produces an egg and a sperm. The X chromosome is active in the egg now, the X chromosome is active in the sperm. These will get fertilized, produce a gamete, or an embryo, with 2 X's. We're looking at females here. This is the embryo, starts to divide mitotically. You get 2 cells. At a particular stage in development, basically at a place in development called blastular formation, okay, when there are about 500 to 1,000 cells in the embryo as it's developing, at that point, at random, one of the X chromosomes becomes inactive. So if you have 500 cells, 200